The next speaker is Valter Longo, who is online. Valter, can you hear us? Oh, here he is. And the title of his talk is Fasting, Mimicking Diet and Cancer Treatment.
Okay, so thank you. Sorry I couldn't be there. And so I'll this is my disclosure slide. And I'll be talking about fasting mimicking diets, and particularly we do a lot of work on them and aging and in normal subjects, but today I'll focus on cancer. And so the fasting mimicking diets were really developed many years ago by my lab in response to the difficulties we had with cancer patients back in 2008, 2009, undergoing water-only fasting. And so we turned to the NCI and to the National Institute on Aging and asked them to fund research to develop something that was equivalent, allow patients to eat, but will have equivalent effects on IGF-1, IGF-PP1, glucose, and ketone bodies compared to water-only fasting. And so we came up with a low-calorie, low-sugar, low-protein, high-unsaturated fat regimen that lasting for mice about 2 days, 2 to 4 days, and for people 4 to 5 days. And so some more of our work on this fasting mimicking diet, or initial work on normal mice, was looking at, for example, the immune system. And this is cycles, 4-day cycles of this FMD, started in middle age. And Sebastian Nin Yang has shown that this can cause a rejuvenation, activation of hematopoietic stem cells, increase in the level of hematopoietic stem cells, and then the starvation, but more importantly, the refeeding, leading to rejuvenated profile, at least in white blood cells and in the lymphoid myeloid ratio. And this accompanying this was the effect on cancer. So in the 57 Black 6 females, this cycle started here about 16 months of age, twice a month for 4 days, leading to a lifelong about 45% decline in the tumors, but also a postponement. These gray dots are the tumors in the control group, and these red squares are the tumors in the FMD group. So a lot less tumors and postponed. And so in parallel with this, we were doing work on cancer treatment. So it's very effective in preventing, let's say, lymphomas and some adenocarcinomas. Could be also effective or make the therapy more effective. And I'm just going to show one initial slide before I move into our newer study, which is the effect of either chemotherapy or fasting and cancer-free survival, right? So what happens if you just give chemotherapy to a mouse with lung cancer? This is actually a study done by a group in Switzerland, and these are our own work. But so either chemotherapy or fasting does little to nothing for cancer-free survival. Of course, it retards cancer growth, but it does nothing to eliminate all cancer cells within a mouse. And the point that I'm trying to make is that when you add the fasting or the fasting mimicking diet, and now this combination of the starvation conditions and the chemotherapy, the targeted intervention, and the wide acting intervention is actually very effective. And now a number of groups have shown this. So what happens? There's two things that happen. One is called differential stress resistance, which is basically normal cells normally are killed by chemotherapy. But when we starve them, the normal cells go into a protective mode, something that we had first described for yeast decades ago. And so there's the differential stress resistance. But then there is also differential stress sensitization, so that the cancer cells, which normally are killed but are not particularly sensitive necessarily to chemotherapy, they're become much more sensitive once the environment becomes much more restrictive, right? And so we look at an example of that. So then we started thinking, so chemotherapy very toxic and starvation protective? Is it possible that we could combine two anti-aging strategies to kill only cancer cells and maybe protect even more the normal cells? So we started thinking about vitamin C. And vitamin C combining with fasting mimicking diet. And vitamin C had been shown by the Kentley lab to be effective against KRAS positive tumors. And so Myra in the lab looked at this. So STS refers to like in vitro starvation, right? So this is in vitro studies. And so the vitamin C, in fact, for these colorectal cancers, lung cancer, pancreatic cancer, is more effective. So STS alone, if it's a KRAS mutated tumors, nothing happens. So fasting alone, nothing happens. And then you give them vitamin C, and you see there is much more toxicity against all kinds of cancers, with the exception of this H23 lung cancer. But the point again is, when you combine the starvation and the vitamin C, now you see this big jump in toxicity by these two effectively anti-aging interventions. So why is that? Well, and by the way, this is also working very well in vivo. So now using this KRAS mutated colorectal cancer, you see here that the fasting mimicking diet plus vitamin C is very effective in delaying cancer progression. And so when Myra looked at mechanisms, this jumps out, meaning that the DNA breaks only in the combination of starvation and vitamin C. Now you see very high level of DNA breaks. And accompanying these DNA breaks, again, with the fasting vitamin C, you see a big jump in reactive oxygen species production. And this is just showing that, by adding glutathione, we can reverse this. And again, the cell death, either N-acetylcysteine or glutathione, or also superoxidase mutase and catalase, most of them were effective in reversing this. So clearly, reactive oxygen species and certainly superoxide and hydrogen peroxide seem to be involved here. And but when she looked deeper, iron seemed also to be involved, and so free iron 2 is increased only in the combination of fasting and vitamin C, and also ferritin. So a decrease of ferritin by the combination of FMD and vitamin C, then leading probably to this increase in the available free iron pool. And so and this is confirmed, the role of iron confirmed by the iron chelator, the spheroxamine. So in every case, this is the toxicity of STS fasting plus vitamin C, and this is reversed by the use of the chelator in 3 different KRAS mutated cancer lines. So we have shown in our previous study on melanoma and breast cancer cells that heme oxygenase 1, which is responsible for the breaking down of heme into ferritin, biliverdin, and carbon monoxide, that this heme oxygenase 1 expression was responsible for the, so decrease of HO1 was responsible for the sensitization of breast cancer and melanoma to chemotherapy. And so when we looked at this HO1, we knew that is differentially expressed. So if you fast an animal, HO1 in the liver goes up, HO1 in the muscle goes up, but HO1 in the tumor goes actually down, right? So there's a differential effects of starvation on normal tissues and cancer tissues as far as HO1 is concerned. And so when we just acted on heme oxygenase 1, this by hemin, which is an activator of heme oxygenase 1, or zinc protoporphyrin, which is an inhibitor of heme oxygenase 1, this was sufficient to, so activate HO1 is sufficient to reverse the toxic effect of fasting and vitamin C. And just the zinc protoporphyrin inhibition of HO1 is sufficient to replace fasting in killing specifically these KRAS mutated tumors. So in summary, what seems to be happening, at least with these KRAS mutated cancers, is that normally vitamin C can be toxic through some phantom chemistry, but by an HO1 dependent vitamin C activates heme oxygenase 1. And so by activating heme oxygenase 1 also increases the level of ferritin, and therefore it generates a scavenging effect for iron 2. And so now the iron 2 is not available to react with hydrogen peroxide and to generate the very toxic hydroxyl radical. In the case of fasting added to vitamin C, now this effect on HO1 and ferritin is reversed, and now this hydrogen peroxide and superoxide are available to react with iron 2 and generate the toxic hydroxyl radical probably. Okay, so then we move this to yet another non-toxic intervention. So we start considering, what about hormone therapy? And so we use this hormone therapy mouse model. And in this case is using the standard of care for humans that you have ER positive, estrogen receptor positive tumors, which is a fulvestrant going after the estrogen receptor, and palbociclib, which is a CDK4/6 inhibitor. And so if you see here, the mice, just like people, with the two drugs are very effective in delaying cancer growth, and then eventually the tumor becomes resistant, and the cancer grows, and the mice die. So when we add fasting mimicking diet, you see that the tumor gets progressively smaller, and some of the mice actually become cancer-free. And after 160 days or so, all the mice are still alive. And the other interesting thing is the reversal of resistance. So even if we allow the tumor to become resistant to the two drugs, and then we intervene with the fasting mimicking diet, then we see the tumor regression, suggesting that the starvation conditions are able to kill resistant cancer cells that have acquired resistance. And this is the interesting part about the starvation and the fasting mimicking diet, is they acting on so many different markers. So in this case, it looked like either insulin, leptin, or IGF-1 were sufficient to cause the growth of the tumors, but all three together were most effective, right? So if we allow the tumor to grow, they apply the fasting mimicking diet, the tumor start undergoing regression. But then if we add any one of these three, IGF-1, leptin, or insulin, the tumor starts to grow. And if all three are present, then the tumor grows the most, right? So you see here the fasting, acting on all three. And of course, it's very interesting now, because it implies that you will need at least 5 drugs just to get this effect, right? So inhibiting estrogen receptor, inhibiting CDK4/6, IGF-1, insulin, and leptin to get an equivalent effect. Clinical studies, in this latest paper, we also had clinical data from 36 patients. And so we heard the criticism for a long time about a patient losing weight and losing muscle mass. And so we looked at this, and if you look at the hand grip, it doesn't change. These are patients that received 8 cycles of the fasting mimicking diet. And actually, when combined with a light muscle training exercise, just 20 minutes, we see that the muscle function measured by phase angle, and also the muscle mass, muscle and bone mass, fat-free mass, goes up, right? So these patients undergoing 8 cycles of the fasting mimicking diet are actually gaining muscle function and mass, and they're losing they're losing fat mass. And then of course, this trial was not going after the progression-free survival or overall survival, but it was looking at IGF-1, leptin, and insulin and ketone bodies. And you see here in both, these are actually 2 trials, one in 12 patients and one 24 patients. And in both cases, you see effects on IGF-1, leptin, and insulin. And in one trial, especially IGF-1 and leptin, the cycles of the fasting mimicking diet are able to have not just acute effects on IGF-1 and leptin, but long-term. So 3 weeks after the end of the diet, the IGF-1 is still significantly lower, and the leptin levels are still significantly lower. So now we've assembled a team of 11 hospitals, and we're asking for funds to do a big trial with 500 patients to see, in fact, if we can change progression-free survival and overall survival in hormone therapy patients. Okay, so this trial was done by Leiden, University of Leiden, Judith Kropp, and Hanno Pell. And this was instead a randomized clinical trial in which patients were either receiving chemotherapy before surgery or chemotherapy plus the fasting mimicking diet before surgery. And actually, interestingly, this received some criticism for low compliance, but this was very interesting to me because in spite of low compliance, if you look at the intent to treat, even so about 50% of the patient completed 2 cycles, and then it went down to about 35% completing 3 or more cycles. But in spite of that, you see that the patients that are not responding to therapy goes from 27% in the regular diet group to 11% in the FMD group, in spite of the low compliance. Now, if you do per protocol, and you look at people that actually did at least half of the cycles of chemotherapy together with the FMD, now this goes from 27% to 5%. That's a 5-fold difference, which I think is remarkable. And I think it's convincing, more convincing when you look at the pathology. So the looking at Miller pain 4-5, so meaning the cancer, so these patients had the tumors removed from the breast, and then the pathologist did the pathology of that. And so the 4-5 Miller pain means that cancers are either 90% cancer-free or 100% cancer-free, so complete response. So you see here, the FMD compliant, this is over 40% that are either 90% or 100% cancer-free, and this goes down to much lower in the regular diet compliant or the FMD non-compliant. Those that were in the FMD group but did not actually do more than 1 cycle or so. Okay, so why does it work? Both for cancer prevention and for cancer treatment is that probably because it's going after so many different markers and some different factors that help cancer stay alive. So glucose, IGF-1, insulin, leptin. Also, I didn't talk about it, but the sensitization of the cells by starvation also is making the attack by immune cells much more effective and much more aggressive. And so probably by changing the environment, making it very complex, and then combining it with a targeted therapy, this is very effective. And so with that, I'm going to end there and thank Mayra Ditano, who did the vitamin C work, and Vanessa Spagnolo, who did the hormone therapy work together with Irene Caffa. Thanks.
Any question from the audience? Morten.
Very nice presentation, Walter. I was wondering if you could, if you're looking into sort of the genotype of the cancer in terms of response to fasting, if there are certain, I guess you have the HER2 negative, but if you look, if this is in your plans to look more broadly into genotypes.
Yes, it's in our plans, but we're certainly looking for collaborations, right? So we, for example, in the Leiden trial, we have samples. Now we're looking at immune infiltration, but I think that there could be analysis on the genome of the tumors and see which one are responding the most and why, right? Is there some particular signature that makes the tumor more responsive, as I've shown for the KRAS mutated, not just colorectal, lung, pancreatic. So, some drugs are obviously very effective against one mutated tumor and not another.
Yeah, one question from the slide. Did you explore what's the impact of FMD on stem cells and how the stem cell niche affected upon refeeding?
We've done lots of animal studies on stem cells, on the gut, on the hematopoietic system, on the nervous system, stem cells, and so we see a clear elevation, particularly in the gut and in the oligodendrocyte precursor cells, etc. The niche, we started to look at the niche. We never published on that. So that would be very interesting. And we just got approved a trial in Italy to look at stem cells in a human trial. So circulating stem cells. And so we're very interested in seeing, are there any that are elevated and which ones are they?
Okay, so no more question. I think we will thank you again.